Carrie Gallagher, General Manager here at Ability Center in Wilsonville. Ability Center has proudly been in business for over 26 years, serving the mobility needs of our communities. We are so proud to partner with the ALS Association and are so proud of the strides that have been made in ALS research. We wish that we could have joined you in person today for this research symposium as we have in years past, but we're happy to be participating in any way that we can. Here at Ability Center, we serve all of your mobility needs. If you have a need for wheelchair accessible transportation, we're the place to go. We have a location in Wilsonville as well as Springfield, Oregon. We sell and rent wheelchair accessible vehicles. So we have used vans starting at about $25,000 all the way up to top of the line vans like this Toyota Sienna van with a Bronability in-floor ramp. We have lots of options for new and used vehicles. We also rent vans. So if you're not ready to own a van yourself and you'd like to have wheelchair accessible transportation for a day or a weekend or a week or however long you need it, we're here for you with rentals available. And as always, ask us about our wonderful buyback program. We are looking forward to seeing you all soon.
Hi, everyone. Welcome. My name is Lance Christian, and I'm the Executive Director of the ALS Association, Oregon and Southwest Washington chapter. I want to welcome everyone to today's webinar. It's the third of 4 webinars this week, Lunch and Learns, focusing on ALS research and scientific and community efforts towards finding treatments and cures for ALS. Today's presentation focuses on Today's presentation focuses on the ALS Association's efforts towards funding coordinated research and strategies for finding new therapies and new treatments for ALS. We'd like to thank our sponsors for helping to make this event and the services and support that the ALS Association offers possible. In particular, we want to thank the VC's Fund, Providence Brain and Spine Institute, Fortis Construction, the Standard, New Motion Mobility, and especially today, the Ability Center, today's sponsor for today's webinar. I'm actually here at the Ability Center in Wilsonville, Oregon, and behind me you may see in a wheelchair accessible van, and the Ability Center is a great resource for families looking to explore accessible transportation options in their community. But before I introduce today's speaker, I want to cover a few housekeeping notes. Today's presentation will last for approximately 30 or 40 minutes with 20 minutes for questions and answers. All participants and video microphones are disabled to reduce interruption. If you have any questions for the presenter during the presentation, please type them into the Q&A box provided at the bottom of your Zoom control panel. Today's presentation will be recorded and made available for later viewing, and today's presentation will also be live streamed over Facebook for people who couldn't register. So I'm excited today to introduce Dr. Kuldip Dave. Dr. Dave is the Vice President of Research for the ALS Association and joined our organization in May of 2019. In this role, he develops and implements the broader research vision and sets priorities for the National ALS Association Research Program. He supervises the operational and staffing and budgetary oversight of our research program. Dr. Dave received his bachelor's degree in biology from Rutgers University, his PhD in pharmacology and physiology from Drexel University College of Medicine in Philadelphia, and he went on to work in the pharmaceutical industry for several years before he joined the Michael J. Fox Foundation for Parkinson's Research. After 9 years at the Michael J. Fox Foundation, he joined the ALS Association to head up our research program. So thank you so much and welcome, Kuldip, and thank you for joining us from the East Coast today. And feel free to take the screen.
Thank you, Lance, and hi everybody. I wish I was there in person. I hope you all can hear me okay. And I'm going to share my screen and let me know when you see it come up. Lance or
Perfect.
Tess?
Perfect.
Everything
We
looks
can see it.
okay, great. All right, so today I'm going to share with you some of our research strategy, and I'll give you a progress update on where we are today in terms of clinical trials. And so before we talk about that, let me spend a little bit time about telling you about our research program. We are the largest philanthropic funder of ALS research in the world. Since Ice Bucket Challenge, we have committed over $110 million to research. If you look at our program today, we have over 170 active research projects that span across 15 different countries. Just last year alone, which is what we call fiscal year 20, we had 70 new projects that totaled over $21.5 million. And I think what makes us different is a couple of things. One, we're not just a funder of research, and you will see the type of research we fund in the next couple of slides. But we also fund infrastructure grants. These are grants to, for example, develop a database so that all of the research data can be stored and shared with other researchers, or developing tools, bench tools, research tools, antibodies, animal models, things like that that can be helpful for everybody. We also fund training and education grants. We have provided funding for training of clinical trial coordinators, of nurses that would run clinical trials. We also fund postdoctoral fellowships and clinical doctoral fellowship grants as well. The more we can get the younger generation, the younger people that are coming into with their degrees to work in ALS, the more likely they're going to stay in ALS research. What makes us different again is the scope, and I'll get into that in a minute. But the mission integration piece, as you know, the ALS Association has a three-pronged mission. Research is one that I'm going to talk about today, but we also have a care services mission and an advocacy mission, and these three mission areas work together, which is different from other organizations in this space. And I think what sets us different is the fact that we're able to leverage our funding. Even though we are the largest funder, nonprofit funder, we are not the largest funder. The National Institutes of Health, that's funded by our tax dollars through governmental funding, funds about $100 million of research each year. And what we found a couple of years ago is that our funding was able to seed larger funding. So $40 million of our funding resulted in $120 million of follow-on funding, mostly from NIH. And that tells you that we have about a threefold leveraging and increasing the impact of our dollars. So before we talk about all the different aspects, I think it's important to understand the ecosystem and the pipeline. What do we mean when we talk about these different aspects of research? So give me about 5 minutes to go through this, because this will really ground you and give you a better understanding of how you actually get a drug to the market. So if you can see my cursor here, all the way to the left in red is the biology and genetics of ALS. This is where you start to ask questions, and really is the first step in this ecosystem. This is where you ask the question, what is different in ALS? What is dysfunctional in ALS? What is dysregulated in ALS? What is the protein or DNA or RNA, or what's happening at our cellular level that changes and that someone gets ALS? And I think that's the question you're asking here. This is the most important step. This is where we do things like gene sequencing approaches, omics approaches. This is what we call target identification. We identify the targets that we want to make drugs against. And that's what happens in this space. Underneath it, this purple part is the risk and causes of ALS. And this is where you ask questions on the environmental risk factors, the occupational risk factors. As you know, serving in the military has been shown to increase the risk of ALS by twofold. And if we can understand what happens in that with those risks and how do they change the biology, we may get a better sense of what drugs to make against those targets. So for example, let's say we find a targeted protein that whose activity is reduced in ALS, then we know that one of the drug approaches could be to increase the activity of that target. And that's what happens here in the second phase called drug development and biomarkers development. And while this on the left is mostly happening in academia, in universities, in colleges, this in green is happening mostly in pharma companies and biotech companies. And they are now starting to develop drugs. This is where chemistry happens, they're screening compounds, they're doing hit to lead optimization, they're testing whether the drug works in animals, whether it's safe in animals. All of that happens in this space in green. And only when a drug is safe and seems to have a signal in animals does it go into this next phase, the blue phase that we call phase 1, phase 2, and phase 3. And this is where it enters the clinic. Clinic means that it's now tested in human beings. Phase 1 is where the drug is tested for safety. Phase 2, whether it's, again, it's tested for safety, but now we start to look at whether it is efficacious or it works in a smaller ALS population. And then in phase 3, where you again look for safety, and now you look for, again, efficacy, whether it works in a larger population. Once a drug passes through this pipeline, it sort of enters into this real world. By the way, this pipeline is not easy for drugs to go through. 98% of drugs that go through this pipeline fail and do not make it into the real world. This is not just true of ALS. It is true of mostly all CNS diseases, whether it's Parkinson's, Alzheimer's, Huntington's, and others. It is very difficult for drugs to get through all of these phases. And this is why the larger our investment in this area, the more shots on goal we're going to have passing through this pipeline, and that something will be successful. On the other side of it, in the real world, let's say a drug does get approved. There's research that we can do on the phase 4 studies. Phase 4 means what is the drug doing in the real world scenario? Now that it's out in the market, how is it behaving? We have research for assistive technology. You've heard about wheelchair access earlier. And we want to make sure that we're driving research in new technologies so that we can make devices available for the patient community. Telehealth medicine, telehealth and telemedicine research into families and caregiver burden, research for access to drugs, access to quality of care, natural history studies, just seeing how the disease progresses, and of course, patient surveys. And then underneath it all is this piece that I talked about earlier, infrastructure, whether it's storage of biospecimens that are collected, data that's collected, tools that we can make available, and of course, training and education research. And underneath that, as you can see, there are public health sciences approaches where we can say, let's say we find out that there's a particular risk factor, exposure to a particular type of toxin that increases your risk for ALS. We can use public science approaches to inform the community so that that toxin is either taken out of our air or our water, or we can educate the public to stay away from areas where those toxins may be more prominent. And so it's important to understand this ecosystem and pipeline. As you see, this is a complicated pipeline, and drug development is not easy. And if you look at our research program, I'm happy to inform you that there's really not much that we do not fund. We fund almost all of the biology genetics part of it, drug development, phase 2, phase 3. And even though we're not able to fund phase 3 studies, there's just about no nonprofit in the world that can, because these studies cost $50 to $60 million just for one trial. We do fund some biomarker components of phase 3 studies. For example, you may have heard earlier this year, we funded neurons, Brainstorm's neuron study, and funded the biomarker component of their phase 3 trial. We also fund research in these areas in the real world, and fund infrastructure as well as prevention approaches. So if you look at our total portfolio, we have currently 175 active research projects that span across the world. And if you count the number of dollars committed to those 175 projects, it comes to $58 million. That's our current research commitment. So let me just make sure from the time perspective, we're doing well. Okay, good. So I think it's important to understand how do research projects get selected? And this is an extremely rigorous, very transparent and deliberative process. And let me walk you through this process so that you understand how projects get selected for research. So the very first thing that happens is we get a letter of intent. We ask researchers to send us a short proposal. You know, this is what we normally call an elevator pitch. It's generally 1 to 2 pages, and it really asks them to summarize their overall idea of their research project. What is it that they're doing? Is it biology? Is it drug development? Is it biomarker development? Is it an infrastructure project? What is it that they're planning to do? Is it novel? Is this a priority for the association? Does it fit the scope of the funding program, and so on? Once a letter of intent is approved, then we ask them to submit a full proposal. A full proposal is now much longer, 8 to 12 pages. But at this stage now, this full proposal is peer reviewed by external subject matter experts, which means that we get people that have professors, industry scientists, people who have the expertise in that particular subject matter. We call them in and we ask them to review that proposal. And the criteria that's looked at is the plan itself, whether the team is the right team, whether the budget is the right budget commensurate of the project, and whether it's even feasible to do something like this in the amount of time that the project has been proposed for. This review committee will now provide recommendations on what can be funded or what should be funded. It's very important to note that the review committee just provides recommendations. The approval for funding is given by the research committee. That's now to the next step here. And the research committee is the committee of our board of trustees. It's volunteers, it's comprised of patients, of caregivers, current caregivers, former caregivers, scientists, clinicians, business professionals. And this research committee is the one that gives the final approval for funding and budget. And once that research committee approves a proposal, it becomes a grant. And this grant project at that stage now, this is in the 4th stage here, which is where now, let's say it's a $200,000 grant. We do not give $200,000 upfront. What we do is we give $50,000 upfront. And then at 6 months, we ask the investigators to provide a progress report. We make sure that the researcher is doing what we funded them to do, and then we release the next 50K. And that same thing happens at 12 months, and same thing happens at 18 months if it's a 2-year project. And this is how $200,000 are given to a researcher. In addition to milestone-based payments, like I talked to you about, we ask the researchers collaborate with each other, that they share data with each other, so that researchers can learn from each other. They share specimens and they publish that data so that the scientific field can learn from the results that are published. By the way, just like I told you earlier about the pipeline and how 98% of drugs fail, if you look at from the letter of intent to the grant project, only about 90% of the projects make it to this through this stage. It's a very competitive process, and you know, this is something that we're very open about, because we want the community to know that we're doing this in a transparent way. Okay, so having said that, I can tell you that 2020, despite the effects of COVID-19 on our fundraising and the limitations that it has imposed on people being able to do research, we have committed over $13 million to 44 different projects. By the way, remember, in the earlier slide, only 90% of the projects end up getting funded through that 4-step rigorous process. And these that you're looking at here are those that were successful to be able to get our funding. The HEALY trial is, of course, run through Mass General Hospital. This is a trial that where you get to test several therapeutics in clinic in phase 2, all at the same time. And so you could test 4, 5, 6 compounds all at once. And this way you save on time. You use the same common infrastructure. And so you save on time and you save on money. And we're very happy to provide and partner up with the Healy Center to fund the platform trial in such a rigorous way. We also funded 5 preclinical projects. Remember the green portion in the pipeline that I was telling you, where small biotechs develop compounds and test in animals? We funded 5 projects like that. And in fact, the Oregon chapter was responsible in helping us co-fund one of those projects. It was a project that we funded that's looking at copper ATSM, which is a drug that's currently being tested in the clinic. This project wants to now take copper ATSM and combine with another drug to see if now with this combination it would work even better in animals. If it does, then it gives us some rationale for taking it into the clinic in a combination product. We funded infrastructure through NEALS. NEALS is a clinical trial infrastructure site that where we provide $650K for clinical trial training, for infrastructure, for patient recruitment and trials. We've also funded 7 projects to the tune of $1.4 million to fund assistive technology projects, brain-computer interfaces, for quality of life research for patients. We funded $500K for the neuron trial for phase 3, where we provided them grant funding to measure certain biomarkers who would better understand how neuron works. And I'm not going to go through all of these, but again, I think it gives you a good sense of the type of projects that we are and we have funded this year so far. So making sure about the time. Good, we have plenty of time. So, you know, as I said earlier, we fund about, you know, last year we funded $20 million of new research, committed $20 million of new research. Yet the NIH funds $100 million of research each year. We know the Department of Defense funds $20 million of research. We know the CDC gives $10 million each year. So we know that there are other dollars out in the field. And so our strategy has been to leverage and increase and amplify our dollar investment. And we do so by many different ways as shown here. For example, on the nonprofit side, we partner up with other nonprofit organizations. So for example, earlier this year, we partnered up with a nonprofit organization, Target ALS. and we provided 350K, and they provided 350K, and we both combined to develop a larger biomarkers project. In similar fashion, we have partnered with MDA, with Project ALS, with I'm ALS on many different scientific projects. If you just added up those up in the last year, we have had almost $1 million external match in these partnerships. The other way we amplify our dollars is that I sit on the advisory committees for the ALS Investment Fund or for companies like Orphazyme. And I'm able to also provide presentations, just like I'm presenting to you today, at investment conferences, to venture capitalists, I speak at least 3 times a week with small biotech companies, and I can provide them guidance, scientific guidance, financial guidance on why they should be investing in ALS space. On the government side, I am now part of the Department of Defense's programmatic panel. As I said earlier, the Department of Defense gives $20 million of ALS research, and this allows me to have a say and a perspective on where those dollars should be spent as well, in addition to our conversations and partnerships with the CDC, the NIH, the VA, for example. And I spoke to you earlier about the Healy trial, but I sit on the foundations committee, and the Healy trial is a $100 million initiative. And again, even though we provided them $3 million, by sitting on that committee, I'm able to have a perspective on that large initiative. I sit on the scientific committee of Answer ALS. I sit on the scientific committee of the International Alliance. And again, these are the ways we can leverage and amplify the dollar. So it's not just directly what we fund, but in these indirect ways where I'm able to shape the dollars that are coming into the ALS field. So now switching it a little bit into our landscape and the state of the field, where are we today? And so this is, since May, and this may have changed just a couple of bit trials, but just to give you a sense of where we are today. Remember I talked to you earlier about phase 1, phase 2, and phase 3 studies? Well, this is just a smattering of some of those drugs that are in each of these phases. The couple of take-home messages in this slide. One, look at the number of different drugs that are in each of these phases. Remember I told you 98% of drugs fail going through this pipeline. And so the more drugs we have, the more shots on goal we have, the better it's going to be for us. The pipeline has not looked this robust in many years. We're just so excited that there are different kinds of drugs. There are small molecule approaches, there are peptide approaches, there are gene therapy approaches, there are cellular approaches in here. There are different pathways that are being affected. Some are going after mitochondrial pathways, some are going after inflammation. So again, really exciting. That's point number one. And point number two is, you can see some of these are small companies that are driving it. Some of these are large pharma. The Healy platform is included in here. And some of the asterisks that you see here with each of these are ones that are ALS Association directly funded either that drug or technology prior to phase 3. And so it shows you the kind of long-term effects our funding can have, even if we may have funded them early on in the biology phase or in the preclinical stages, that it can have an impact of them moving into the clinical phase. Just to tell you some of the good news here or what to look forward to, I'm going to talk about AMX35, which is right here, in a minute now. But you have heard earlier this year that tofersen Biogen showed data and published data that tofersen was shown to be safe in an early study clinical trial and was shown to reduce SOD1 levels. You also know that Neuron is expected to read out its phase 3 study by the end of this year. And of course, I talked about the Healy trial a little bit earlier. So what's the state of the field? Let me just make sure on the time. The state of the field is good. We have, let's start from up here in blue. We have a really good understanding of ALS. There are huge gene sequencing approaches that are going on, a lot of them funded by us. We are funding things like CREATE, Answer ALS, Project Mine, New York Genome Center. Again, the idea is that the more gene sequencing we can do, the more targets we may be able to find through these different approaches, omics, epidemiology studies. Going now to the one in the red here, we have diverse biological targets. I showed you in those slides earlier, you know, there are drugs that are targeting C9orf, there are drugs that are targeting SOD1, there's Tregs, the cells that are being developed by Stanhope Health, tyrosine kinase, urate, and so on. There are multiple drug approaches. Again, as you saw in that earlier slide, there are chemical compounds, small molecules, there are antibody type approaches, there are antisense oligonucleotides, there's stem cell approaches, AAVs. Again, you don't have to know all this. The point is that the more shots on goal, the more diversified our portfolio is going to be, the better it's going to be for us. We have institutional engagement now. There's, you know, Merit Sikovich from MGH always likes to say we have over 100 different companies that have either a preclinical or clinical program in ALS. We have clinical trial efficiency. We know last year the FDA provided guidance to sponsors as to how to run the trials and what kind of outcome measures to include in their trials. And of course, we have community engagement, whether it's other nonprofit organizations that work together, or the CDC that has a registry where they count the number of people, or people like yourself that are joining today and want to hear about what's going on. Okay, so in the last 10 minutes, I want to talk about AMX35 because I think you've all heard a lot about this, and I just want to make sure that you understand where we are today. So you heard, this is the trial that we funded. This was one of the first studies that we funded through the Ice Bucket funds. And it's this drug called AMX35. It's a combo product that's being developed by the company Amylyx. And what their drug trial showed, Centaur trial showed, which was, by the way, a robust and well-designed trial. It was double-blinded, it was placebo-controlled and multi-center. There was a huge study, 137 patients across 25 different sites in the United States. And what they showed was that AMX35 significantly delayed ALS disease progress. So, you know, over a 6-month period, it showed a 2.32 point difference. And if you looked at it a little bit differently, it changed from baseline. It had a 2.92 point difference over 6 months. And this change of a 30%, it can be very clinically meaningful for, you know, for an ALS patient. This could mean someone being able to, you know, feed on their own versus needing a feeding tube, or being able to walk on their own versus needing a wheelchair. This effect was on top of the standard of care. So most patients that were taking AMX35 were also taking Riluzole or Radicava. So the drug actually had its effect on top of that, which is very significant. And the secondary endpoints were also in the same direction. What's most important is that AMX35 demonstrated an extremely good safety profile. It's really well tolerated. There were some adverse events that were gastrointestinal related, but they were transient, short-lived, and went away as the trial went on. On top of the efficacy results and safety results, Amylyx also looked at survival data, and they followed patients up to 3 years in this trial to look at median survival. And what they showed was that AMX35 demonstrated a 6.5-month increase in survival in participants that were originally randomized to the AMX35 group versus those originally randomized to the placebo group. And, you know, if you looked at it a little bit differently, if you looked at the lowering of the risk of death, AMX35 lowered the risk of death by 44%. So again, this was the first drug in decades and many years that have achieved both a positive benefit in both function and survival, that these effects were on top of, but independent of baseline use of riluzole and or radical. And so what would happen generally, let me just make sure of the time, good. What would happen generally is in a standard approach, because this was a phase 2 study, the FDA would ask Amylyx to conduct a larger and longer trial. That would mean more patients, more clinical sites to test for safety and efficacy. Remember, I told you phase 1, phase 2, phase 3. Well, what I showed you was results from phase 2 study. And normally FDA would say, now test it in a larger phase 3 trial. And this phase 3 trial could take, more than 3 to 4 years. I mean, think about the phase 2 that I just reported data on. That trial started in 2016, and we're looking at data now in 2020. This phase 3 trial, which would need more patients and would require more time than 6 months, would be, include site selection, activation, IRB approval, recruitment of all those patients, data collection, data analysis. We're looking at a good 3 years at the minimum. And even if it is positive after this 3 years in a phase 3 trial, now the sponsor Amylyx would need to prepare a new drug application, which sometimes can take 6 months because these applications can be 1 million pages long. And then the FDA reviews that new drug application. And if they approve it, even that can take up to 6 months. And so we're looking at a minimum of 4 years before Amylyx would be available. And what we're saying is that, look, if the larger trial confirms these results, that's great. But now the ALS community would have waited 3 to 4 extra years to have AMX35 as a treatment option. It is possible that maybe what we are seeing now in phase 2 is a fluke, is a false positive, and maybe the phase 3 disproves these results. Well, if that happens, then many people with ALS would have probably taken a drug with very little benefit, but also very little harm. And what we're saying is the risk-benefit ratio, the benefit-risk ratio for this drug is so much better that we think that it is worth the risk of skipping phase 3, and that this will allow ALS patients to have access to the drug today. So what we're asking is a proposal to save 36 months by asking FDA to have AMX35 skip phase 3 and get to the ALS community faster. We are asking the sponsor to provide expanded access to AMX35 while the FDA takes a look at that NDA now. And we're asking the FDA to quickly approve with, of course, conditions so that there could be post-market studies where Amylyx could determine the benefit of AMX35 in a larger population in the real world, as I told you earlier on. And so here's the petition that you can sign, or you may have seen, where you can ask the FDA to do all of these things so that AMX35 is available to the patient as a treatment. We're not saying it's going to work in every patient. We're not saying that the phase 3 results will be the same as phase 2 results. What we're saying is that the risk benefit based on the results we've seen so far is so positive that it should be available as a treatment option. And if the doctor thinks that that's the right treatment option, that the patient should have access to it. And so this is where I'm going to end. And this is the pathway that I talked to you earlier, which is sort of the standard pathway. It would take 3 to 4 years to access. What we're asking the FDA is to approve this drug now. And, it would still take a year, but at least it would be available in an accelerated fashion. So I just want to stop here and say thank you, and I'd be happy to take your questions after this.
Thank you so much, Dr. Dave. We appreciate your time and your overall explanation of the research pipeline and the timeframes it takes to move science into finding actual treatments that can be available in the clinic. I particularly appreciate this information because as a chapter leader, I want people to understand and know that our efforts to fund research across the association are collective. All the dollars that we raise here in Oregon are part of this national effort that funds both international research. And it is truly the experience where all of us together funding these efforts are more powerful with a coordinated effort led by our research committee and Dr. DeVay. So thank you so much. We appreciate your energy and effort. So
Thank you, Lance.
questions that have come in, and I just want to summarize a few of them. We've had a couple questions coming in asking questions about studies of environmental exposures as causes for ALS. And in particular questions, things about like radiation studies of people who have maybe worked in the airline industry, or even simple things like mercury exposure from broken thermometers. Does the ALS Association fund environmental aspects related to ALS, and are there any interesting findings in those realms?
Yeah, thank you for that question. This is actually a really right time and exciting time to ask that question. And I'll tell you in a minute why, but let me answer your question first on, do we fund these type of studies? Yes, we do. We've funded studies into environmental exposures where currently, for example, I'll give you an example of a study we're funding now that is looking at blood samples, and within blood is looking at what kind of exposures to toxins people have had over the years, and correlating that with whether that person ended up getting ALS or not. And so this way we can correlate a particular toxin and a threshold of that toxin to increasing your risk of getting ALS. Similarly, we have funded studies to correlate this other study that's looking at exposure to lakes. So there's this idea that green, blue algae in lakes and the toxins that the green, blue algae are spitting out into the lake area may increase your risk of ALS. And so we're funding research both in Florida and New Hampshire, looking at how close you live to a lake that may have that particular green, blue algae and what kind of exposures are needed. Now, it's very important to understand there is no straight line you can draw from a particular environmental exposure. Not everybody that serves in the military gets ALS. Not everybody that is in airline industry gets ALS. Not everybody who lives around a green, blue algae lake gets ALS. ALS is a fairly rare disease. And so this is why, if you remember my pipeline, Lance, that early on part where biology was, we think what's happening is that we're all exposed to a particular risk factor, but there's something within our biology that makes us more susceptible to that environmental risk factor than somebody else. And so I think that's the type of studies we're doing currently, where we can correlate environment and biology. What's that gene environment combination that we need to do?
And that leads into sort of the next question that people have been asking is, what studies, what's being done in terms of studying genetics? Because it sounds like there's this interaction between environments and genetics. And what kind of studies are happening in the genetic realm that's going to lead us to potential new treatments?
Yeah, so huge studies, the gene sequencing approaches, and we have been the leaders in this. I think I talked to you earlier about the type of gene sequencing approaches that the ALS Association have supported, whether it's Project Mind, whether it's Answer ALS, whether it's New York Genome Center, Create Consortia. We are genotyping thousands and thousands of ALS patients and trying to identify gene mutations that are different. And then the ideal part would be, Lance, to then take that information we have on gene mutations and overlay that with the environmental risk factor research and see if there is a gene environment interaction that increases your risk or lowers your risk of getting ALS. And so Again, tremendous research happening. Just to give you a sense of our dollars and our direct funding for gene sequencing has allowed discovery of at least 5 new genes in the last 5 years. And there are already drug companies that are exploiting those genetic targets for therapeutic targets.
Which brings a question for me, Dr. Dove, which is, if you look at the dollars that we invest from the ALS Association, are those efforts kind of multiplied through these collaborations and then sort of leading into industry investment?
Yeah, and we have seen that, Lance, over and over. So a couple of studies I'll talk about here. This is a great question, right? Because we know that there's industry that's funding ALS. We know that there's government that funds ALS. We know there are other nonprofits, but it's still small. Our $20 million is still, not enough. There's never enough money. And so how do we amplify the effect of our investment? And that's what this study that I talked about earlier, where $40 million of our funding to researchers, they were then able to capture data and then use that data to apply for more funding, whether it was from industry or from National Institutes of Health or from other nonprofits. And so we saw that $40 million resulted in $120 million. And the word I like to use is seeding. We were able to seed that funding, that 3 times leverage. It's extremely important in a field of biomedicine.
Yeah. Which really speaks, yesterday we had Dr. Nick Olney talking to us from the Providence ALS Center, and he showed the large increase in the number of studies going on in scientific publications and clinical trials, which sort of speaks to making it harder when there's a lot more activity. How is it, what's the strategy for the association in terms of which projects to fund? Are we able to fund some little riskier or more novel things as seed money versus needing to find an immediate outcome?
Yeah, that's a great question. And what makes our program so different is that, we even though we have that rigorous process set up, or 4-step process that all of you now hopefully appreciate and understand, what that process is doing is able to identify novel out-of-the-box ideas. I mean, the Healy trial, it's never been tried before. The platform trial has never been tried before in ALS. And it's a risky paradigm. We don't know if it would work in ALS. But it survived that 4-step process whereby we were able to fund them in a fairly rigorous way, $3 million over 3 years. But that's a large project. We have funded smaller projects. I'll give you an example. We funded a $50 million, $50,000 of a grant, a fairly small grant that was looking at a particular pathway, cellular pathway. And this is a pathway in oncology, actually. It's in cancer. It's been studied in cancer and never studied in ALS or Alzheimer's or Parkinson's. And this group was able to get data on that pathway and showed that this pathway that nobody ever thought was involved in the central nervous system, everybody kind of associated it with cancer, was indeed associated with the central nervous system. And because that study was able to do that, now we had oncology drugs against that pathway. And so pharma companies can now take those oncology drugs and see if they would work in ALS, right? And so that's the type of out-of-the-box ideas that we want to make sure survives our project selection pathway, because science is complex, and there's serendipity built in, and we just don't know where we'll find success.
Yeah, I appreciate you sharing the number of work groups and committees that you sit on across a wide variety of both private industry and government sort of efforts looking at ALS, because this cross-pollination, it's hard for everyone to keep track of everything going on. And do you feel like the ALS Association and your team are able to sort of be the hub for that?
Yeah, and again, being the largest funder of nonprofit research comes a responsibility, right? We set the example for the community, and if we are transparent, if we are collaborative, if we partner and synergize in an effective way, that's going to really drive the entire field. And so, these committees that you talk about, Lance, are so important. I'll give you an example of this. I joined in May of 2019, and the research team was actually thinking about a biomarkers program to launch. And by sitting on and having conversations with Target ALS, which is a much smaller nonprofit organization than the ALS Association, but in talking to with their CEO and sitting on their committee, what I saw was that they were also developing a biomarkers initiative. In fact, they had spent a year developing a biomarkers project, had vetted the scientists, had vetted the science, had reviewed the project, had made it all, shaped it in the right way. And, it made sense at that time that instead of doing the same thing and sort of reinventing the wheel, it made sense to just take some of our funding and provide that funding and partner up with them so that they can expand their efforts. We don't have to reinvent the wheel, and we can actually maximize the impact that the dollars of two organizations can have. And so this is the type of synergy that we're going to require I told you this pipeline is very complex. It's very difficult for drugs to succeed. And so the more of this type of, rowing the boat in the right direction, in the same direction, the more of that we can do, the better.
Great. A follow-up question to your presentation. Thank you for sharing the rationale for the excitement about AMX, the Amylyx AMX 35 treatment, and the efforts to sort of push early access for people to be able to get that treatment. One of our viewers asked the question, have there been other examples where petitions like this at phase 2 have moved a therapy forward?
That's a good question. I'm not sure, Lance, about that. The problem a little bit is that when petitions go in to the FDA, but the process at the FDA is closed out. So we can see what gets approved at the end, but we can't see what's happening in between, right? And that's something that happens that the FDA and the sponsors kind of interact with each other. Now, the couple of good things here. One, Amylyx has asked us to be part of the conversations when they go and speak to the FDA, which is great because we get to hear and bring the patient perspective in those conversations, that it's not a conversation of just a regulator like FDA who's approving and the corporate sponsor that has the drug, but that there is a patient quotient built into that decision. And I think that's going to be very important. Look, I said this earlier, Lance, this is not an easy decision for the FDA. There's a reason why this is set up the way it is set up, because they want to make sure that patients are not exposed to drugs that, don't work. And God forbid, if they're not safe, now you've added additional burden on patients. And so it's not an easy decision for the FDA to take. And we are cognizant of that, but we also think that this drug, unlike any other that we've seen in the field yet, with its safety and benefit in both function and survival, I think is the results are just so robust that we think that this petition is necessary.
Great. We're getting close to the end of our time, but I wanted to ask you one final question, which is, I know you can't share projects that are going to be funded yet, but I guess I just want to ask you, are there things that you're excited about that are going to be happening in the year ahead? And any interesting new publications that are going to be out, or study or efforts that are going to be funded that you're excited about?
Yeah, so you're right, Lance. I can't talk about individual projects because I have under confidentiality on them. But let me tell you, I think what you're giving me is an opportunity to talk at the highest level what I'm really excited about. So you asked me earlier, Lance, about gene sequencing approaches, and we funded these multi-million-dollar approaches, huge approaches, about 5 or 6 of them across the world. And those have now matured. So they are now undergoing the process where they're looking at what genes are changed, what mutations are prominent in ALS. And we funded, for example, Dr. John Landers and Jonathan Glass last year to now take all of this genetic data from different data sets and put it all into one big data set, and now make that data set available to the entire world. That really excites me because, now you're combining not just you're not looking at 5 individual studies. Now you're combining those 5 individual data sets into a larger data set, and it will be open to everybody for free so that people can go in, they can do their own analysis, their more iterative work, and hopefully we can find more gene mutations and cellular mechanisms so that we can then have better drugs that are made and that can enter that pipeline where you have that kind of 2% success rate. And so that's what gets me really excited, that we're at that stage where I think 2 things. One is that, and the other thing, Lance, that makes me really excited is you saw the pipeline of phase 1, phase 2, phase 3 studies. Neuron's going to read out at the end of this year. Orphozyme study will read out next year. Biogen study will read out next year. And, we just think that these trials have learned from earlier biology. They've really built on better studies. The Healy trial is now going and cranking out and testing clinical therapies. We just hope that we get to see more positive results like we saw with AMX35 come through the pike.
Yeah, thank you so much, Kuldip. I want to share, yesterday was actually my 18th year anniversary of being with the ALS Association.
Congratulations.
Thank you. And I am so excited about the state of ALS research and the progress being made. 18 years ago, I think even a glimmer of one of the things that you just mentioned would have been a successful year. And now it's one thing on top of the other. The amount of energy and effort, it's just increased exponentially over these years. And the efforts to improve and support people's quality of life and trying to get through innovative programs like the Healy trial, access to more and potential new treatments to people faster. I think there's great energy going on, and it's really exciting. So thank you for your efforts in all of this and for coordinating the research strategy. On behalf of everyone at the ALS Association Oregon and Southwest Washington chapter, I want to thank everyone for supporting our efforts. Appreciate Dr. Dave mentioning that our chapter has consistently contributed above and beyond to the national ALS research effort, funding particularly copper ATSM with Dr. Joe Beckman and Dr. Peter Crouch, and all of your support for our chapter also helps with the research mission. So we want to thank everyone for joining us today, and thank you again, Dr. Dave, for presenting and giving us an hour of your time today. Join us tomorrow for our Lunch and Learn. Dr. Sabrina Paganoni from the Healy Center at Mass General is going to join and talk a little bit more, particularly about the platform trial that was mentioned today. So thank you so much, and we appreciate your time.
Thank you, everybody.
Thank you.